Hi everyone, my name is Jared Stevens, and today I'm going to be presenting on my senior thesis study, in which I look at how the acoustic behavior of common bottlenose dolphins changes with group size and the presence of young. Now, to give a little background, bottlenose dolphins display complex acoustic abilities, allowing themselves to produce numerous sounds. We also know that they have a wide range of social behavior, and this behavior is often influenced by a variety of factors, such as physical and environmental factors, as well as social, and that could be based on the type of dolphins as well as the number of dolphins that they're around. Now, from an acoustic standpoint, we know that bottlenose dolphins produce three main sounds: echolocation, which is used mainly for navigation and foraging; whistles, which are used mainly for communication; and burst pulses, which are also used mainly for communication. Now, the goal of my study is to determine how the acoustic behavior of common bottlenose dolphins down here in southern Florida changes based on three factors: how the presence of calves relative to group size, presence of juveniles relative to group size, and then just the overall group size as well. So when we were in the field collecting our data, we used a calibrated digital acoustic recorder and hydrophone and towed it behind a boat in front of dolphin groups. This is how we collected our sound files. Now, while we were conducting these boat tows, we were also doing above water surveys in which we calculated and documented the group size and group composition of these dolphin groups. And then we correlated each sound file with each documentation, and that's how we combined our data together. Now, each of these boat tows was conducted in Boca Chica Bay, Tampa Bay, or the Gulf Shore waters off the coast. Once we collected our sound files, we brought them back to the lab, and we used the spectrogram software Raven Pro, which was developed by Cornell University, and we looked at the first 15 minutes of sound file that had high signal-to-noise ratio, pretty much where there's a little masking that could hide dolphin sounds. We used 10 24-point spectrogram views, and we were looking for three main sounds: echolocation click trains, whistles, and burst pulses. Now, these are examples of what the three sounds look like on the spectrogram view. The first one being echolocation. These are broadband pulsed clicks, and we often found these in large groups, in which we would call them click trains, where we saw numerous clicks following one another. The second one is whistles, and these are more narrowband tonal sounds, and we tended to find whistles either by themselves or just in small groups, just a couple. And finally, the third one is burst pulses. Burst pulses are similar to echolocation, in which they are produced with a wide range of frequencies, except that burst pulses have such a high pulse rate that on a spectrogram view, they come up as horizontal bands rather than vertical bands like echolocation. So what do we find? Well, after analyzing 13 dolphin groups, we used generalized linear models to analyze our data. We found three significant relationships from these tests. First, we found that as juveniles relative to group size changed, we also saw a change in echolocation production. We also saw that as the overall group size changed, we also saw an echolocation production change. And finally, we found that as the calves relative to group size changed, we also saw a significant change in burst pulse production. So we took these three results and we graphed the significant relationships. And we found that as group size increased, echolocation rate decreased. As the number of juveniles relative to group size increased, the echolocation rate increased. And as the number of calves relative to group size increased, we found a decrease in burst pulse rate. But what exactly does this mean? Well, looking at the first point, we see that the decrease in echolocation rate with an increase in group size could be due to eavesdropping or just the general social behavior of bottlenose dolphins. A previous study found that as the group size increased, the bottlenose dolphin groups tended to echolocate less. And this study believed that this was due to eavesdropping, where other dolphins were picking up the echolocation from the dolphins around them, which required these dolphins to echolocate less. Now, we also believe that the increase in group size could also have an effect on the echolocation rate, because these dolphins are very social animals. And so as the group size increases, these dolphins are probably interacting more with one another, which requires them to echolocate less. Now, our second point, looking at the increase in echolocation due to the increase in juveniles, we believe this is due to the natural, independent, and curious nature of juvenile dolphins. these juvenile dolphins are young, in their teenage years, so to say, and so they're kind of figuring out the environment around them. We often saw this in the field, where juvenile dolphins tended to be the dolphins that came closest to the boat and closest to the hydrophone. We believe that this is just the natural inquisitive nature of these juveniles, and that they're kind of testing out their independence, as well as just kind of figuring out the environment around them. And so we believe this is why we see an increase in echolocation with an increase in juvenile dolphins. Now, third, we expected to see an increase in burst pulse rate with an increase in calves prior to the study. And this was based on a previous study that was done that found that mothers tend to burst pulse more around their calves and use it as a form of communication to their calves. And this is entirely due to the fact that burst pulses are produced at such a high frequency. Most predators and other marine animals can't pick up these sounds. But we, however, found a decrease in burst pulse with an increase in calves, which kind of goes against what we believed would happen. But that just leads to the fact that we need to conduct more studies and kind of figure out why this is. So what's the importance of this data? Why is this important to know? Well, first off, these findings will help us increase our knowledge of Varner's dolphin acoustics. But this knowledge will then help us better develop autonomous acoustic recorders. These autonomous acoustic recorders use AI technology and self-learning technology to recognize and document dolphin noises. An example of this is the Haiku Marine Station, which was just put in at the Galbraith Pier at Eckerd College. The Haiku Marine Station and the AI program that's in it will be able to recognize dolphin whistles that come through the channel and upload them to a cloud-based server. This will help the scientist, or Dr. Pierre Simard, who is in charge of this project, be able to document dolphin whistles quicker, rather than scientists having to go through and manually count whistles in each sound file. And these autonomous acoustic recorders in the future will be able to help us calculate population estimates. And these population estimates will then hopefully help us in conservation and policymaking, and will help us better understand the dolphins in Tampa Bay, as well as what we need to do to better our lives around them. So from here, this study is still young. This is a recent study, and so for future research, we need to increase the sample size. This will allow us to get more detailed results and better understand the significant relationships between these different variables and the acoustic behavior of common bottlenose dolphins. We'd also like to see more specific group composition, as well as more detailed acoustic recordings of these dolphins, meaning we'd like to see more groups of each individual group size, as well as calves and juveniles, as well as we'd like to see more detailed acoustic recordings of which dolphins are making which noises. I'd like to thank Anya Cervantes for her help on this project and going through sound files with me. I'd like to thank Eckerd College Dolphin Project for the volunteers and staff that allowed this study to happen, as well as the Eckerd College Natural Science Summer Research Program that provide the funding for my research. Thank you very much.